Today it's my pleasure to welcome a friend from the National Institute on Drug Abuse, Lud Cadet. He's a neurologist, a psychiatrist, and his area of research is he's on the National Institute of Drug Addiction, so it's on drug addiction. And he's done a lot of work, most recently, a lot of focus on psychostimulants, particularly methamphetamine. So I think methamphetamine will be kind of one of the main drugs of abuse we'll talk about, but there's others that do similar things. We'll probably incorporate it in. So Lud, it's nice to see you again. I actually haven't seen you since I retired 3 years ago, I don't think. And
yes, that's true.
First, can you talk about your upbringing? Because it's a little bit not typical for most neuroscientists and neurologists in this country. So you were born in Haiti. Take it from there.
Yes. Thank you, Mark, for inviting me to do this. And indeed, it's a great pleasure to see you again. We're not walking through the building and seeing each other as much as we used to since you left. So in terms of upbringing, I was born in Haiti. I spent the first 17 years in Haiti where I went to Catholic schools. And so I can tell you that I became interested in medicine since I was 4 years old because my mother actually was initially a nurse at the hospital. And then in Haiti, you can progress to to other field, and then she became the head of a pharmacy at a hospital. So during that time, I went to people consider one of the best Catholic schools in my town in Cape Haitian. And when we were there, my parents.
This is in the 1970s. Then is it?
That was in the 1960s. I came to the United States in 1970.
And so you grew up in Haiti under kind of an authoritarian regime, Papa Doc and Baby Doc. And
yes, I was going to talk about that. So actually, I grew up under Papa Doc, and then there are these people who work for him called the Tonton Marcouts. So all of us were very afraid of the dictatorship in Haiti. So for me, actually, whenever I see anything going on in the United States that resemble authoritarianism, that scares the hell out of me. Yeah. So because it was very threatening. So it was important for us to leave because at that time, if you talk too much, there's some things you wanted to do you might not have been able to do. So we came to Brooklyn, New York in 1970. I went to high school for about 6 months or so. The people running the high school say I didn't belong in high school. I was 17, so they graduated me in 6 months. And I went to Brooklyn College. And from Brooklyn College, I went to Columbia University for medical school. And as you mentioned before, I trained in psychiatry first at Columbia University, and then in neurology at Mount Sinai Department of Neurology, and did a fellowship at the NIMH, was an assistant professor of neurology and psychiatry at Columbia before coming to the National Institute on Drug Abuse. But the time in Haiti, I tell a lot of people, the time in Haiti, working around a lot of very smart people was what propelled me to want to go to medicine and actually pursue the dream of becoming a scientist.
And so when you moved to the United States, then did both your parents did your mom continue to work?
No, actually we all came. So one of the troubling thing about it is that my parents didn't speak English, right? So my father ended up working now in a factory, and my mother was like doing odd jobs around Brooklyn, but she never was able to go back because they were already in their 50s. Yeah. They were never able to go back to the same type of upper middle class life that they had in Haiti. So it was very stressful for them, but they felt that the best way for our future is to actually, so they were doing it for the children, leave a place like an authoritarian dictatorship, get out of.
You went to medical school at Columbia, and then you did a residency there too. And Columbia, for people who don't know, that it's located north of Central Park and right by Harlem, right? It's kind of,
that is correct,
borders on Harlem. So you probably saw a lot of.
So I can tell you about when I was a medical student in New York in the 70s, mid 70s, I started in 1975. Around the time, at that time, the most common drug that people were using, for example, was heroin. And so we saw a lot of heroin users in the streets. And then I did rotation at Harlem Hospital because Harlem Hospital is affiliated with Columbia University. And so we saw patients with all kinds of medical issues. That's actually when I became aware of health disparity by working at Harlem and working at Columbia Presbyterian and seeing the differences in terms of what people showed up with and the care they got at a hospital that was a public hospital and the care that people got when they were in a private institution like the Presbyterian Hospital.
Yeah, it's still a big problem across the country. I mean, we see it all the time. COVID was a good example, but it doesn't really matter what, people in the inner cities and the poor neighborhoods that are struggling, they also don't get the health care oftentimes that they need.
They don't.
And it's true in Baltimore where we are now. I should mention that, so the National Institutes of Health, people may think of that as being in Bethesda down by DC, but two of the institutes, the National Institute on Aging where I was and the National Institute on Drug Abuse where Jean Lud is, are in Baltimore on Johns Hopkins Bayview campus. And so Jean Lud, I think you have an adjunct appointment at Hopkins still, right?
Oh, I have actually, mine is at University of Maryland.
Oh, yours is at Maryland. Mine is at Hopkins.
At Hopkins, right.
But as we talk about this, your work at NIDA, Hopkins is important, right? Because they have a facility there for drug addicts, and actually maybe towards the end of the talk we'll maybe even mention psychedelics, because I wanted to ask you some things about that in regard to drug addiction. But anyway, let's go talk about your research on psychostimulants. And so what are psychostimulants?
So psychostimulant, I guess in terms of what I study mostly, I studied the amphetamine type psychostimulants that include amphetamine, methamphetamine. But other drugs that are considered psychostimulant include nicotine, also cocaine, right? But my focus has been only basically on methamphetamine. I've published some papers, clinical papers on cocaine, and I've collaborated with some investigators in the past on cocaine, basic science aspect of cocaine. But almost all of my work has been on methamphetamine use disorder. And the reason why I picked methamphetamine, it's because methamphetamine is reported to only be second to marijuana in terms of drug that are used throughout the world. There are about 30 million individuals that have used amphetamine type stimulant. And in the North America, there's a high prevalence of methamphetamine use from people between 15 till about 64. So it's a very common drug. During the time when we all were talking about the opioid epidemic, people overdose from opioids and everything like that, there were also quite a few individuals who were overdosing with methamphetamine. So that story kind of got buried because the opioid problems was so prevalent. Then there's some report that between 2007 to 2018, the number of overdoses increased by 7.5 in the United States. So it's really very, very important to try to understand the basic neurobiology of methamphetamine use disorders. Sadly, I have to say, at present, there's no FDA-approved medications to treat people who suffer from amphetamine-type stimulants. So it really behooves us to try to come up with ways of addressing the lack of treatment, because that leads to a more clinical complication, psychiatric complication, neurological complication, cardiac complication, and we need to be able to do a better job for our patients.
Yeah, and I mean, you see with cocaine a lot, you occasionally see someone die because it can kind of increase blood pressure. It's a stimulant, right? So
right.
that not only does your brain more active, but your autonomic nervous system, sympathetic nervous system is more active. And so, I did methamphetamine, the molecule itself, where does it come from?
So that's an important question. The methamphetamine was synthesized by Japanese scientists in 1919. And they did that by reducing pseudoephedrine, right? Mm-hmm. Which is, you can find that in plants. And so there's a ephedra family of plants that have these ingredients in them. And it's really basically a simple reaction to make amphetamine and methamphetamine. So after methamphetamine was synthesized, it was marketed, right? Okay. And then it was legally available, and it was marketed like Dexosin. It's DEXOXYN by Abbott Lab, right? And it was later on sold as methadone, and it's still being actually sold, and it's used right now. Presently, it's used mostly with children and adolescents with attention hyperactivity disorders, right? So those kids who don't respond well to Ritalin, okay, so often the psychiatrist or whoever is taking care of them, pediatricians, might opt to use one of the amphetamine type medications. So but it's scheduled, only certain physicians with a specific DEA license.
Because that's kind of scary, right? Because it has potential for addiction.
So actually, for kids with ADHD, the literature says that it's more likely that people with ADHD to develop addiction if they're not treated. But if they're treated with the dose of drugs that they're treated with, it's quite unlikely that they'll develop addiction or substance use disorder. But the one thing about that, it's like brothers and sisters and other people in the family might sell the drug so it can get diverted to illegal use. So that's an issue that parents and other people who deal with kids with attention deficit disorder have to pay attention to, the diversion to use.
And not all people that take methamphetamine or cocaine or opioids become addicted, right?
Yeah, okay, that's a very, very, very important point also. So when I mean, you're old enough to maybe remember that there was a time, I guess in the 70s or 80s, where people were showing pictures of the brain, normal brain, and the brain of on drugs. And then they used to sort of fried eggs.
Yeah.
Is what the drug does to your brain, the drugs do to your brain. But it's actually not necessarily the case that everybody who get exposed to this drug, would take these drugs, would develop drug addiction or substance use disorder, which is what it's the preferred term. So if you think in terms of the numbers themselves, for example, for tobacco, right? Only 24% of the people who use tobacco actually become addicted, quote unquote, to tobacco. For heroin, it's between 23 to 40%. For cocaine, it's about 17%. But for marijuana, it's only about 9%. But for methamphetamine, it's been reported that 40 to 50% of the people who abuse methamphetamine might develop a psychiatric disorder that we label methamphetamine use disorder or methamphetamine addiction. So it seems that of all the drugs, methamphetamine might be one of the drugs that most, like the majority, it's not the majority, but close to the 50% of the people who use methamphetamine might develop a methamphetamine use disorder by psychiatric terminology, right? You've heard about the diagnostic statistical manual. And then, so there's some criteria that people have to fulfill before you can make the diagnosis. So somebody could be using the drug on a regular basis, they work, they don't get into any kind of medical or legal problems. So they won't meet the criteria for methamphetamine use disorders. So it's very important to keep that in mind, because the brain, as you know, responds differently based on which your genetics, your environment, and how much of the drugs that you have been taking.
So the drugs of abuse can be studied in humans, obviously, and also in animals. Now, in humans, what happens, for example, you can look at neural network activity in the brain by functional magnetic resonance imaging. What happens when someone takes methamphetamine? I guess you can snort it, or you can inject it, or I don't know if you can take it orally and it has any.
Yes, actually, there are different ways of taking the amphetamine and also type drugs. Usually, people will start via the oral route, right? They start with a dose of the drugs, and those people who are going to become addicted later on might progress to taking more because they develop tolerance to a small amount of the drug, right? So over time, they take one pill, then it's two, it's three, and then they might progress from the oral route to, say, the snorting route, right? And then after snorting, they might go to smoking. And then most of the people who meet criteria from what I've read, they use it through the IV route, right? So they intravenously, they inject the drug, and I think they're thinking that they're going to get a better high, and then you can inject more drug than snort drugs or taking drugs by mouth. As you become more tolerant, your dopamine system becomes tolerant. It takes more drug to release dopamine, for example, and glutamate. So they increase the intake. And then the effects of the drug last differently depending on who it is who's taking it. So the clinical presentations of the methamphetamine in people who take methamphetamine, usually within the first 2 hours or so, people become euphoric, they become very aroused, their heart, which is a problem, the heart is beating really fast, they have high blood pressure, they can have heart rhythm abnormalities, and all of these you get. So the brain is being affected, the cardiac system is being affected, and this is due to the fact that the drugs are not only releasing monoamines like dopamine, norepinephrine in the brain, but it's also releasing dopamine, norepinephrine, and also epinephrine in the peripheral organs like the heart or the arteries. So it's caused constriction of the arteries. And so when that process occurs in the brain, it can lead to strokes. So some people have ended up in emergency rooms because they might have a methamphetamine or amphetamine-induced cerebrovascular accident. So that's something that people have to keep in mind if they're thinking of taking large dosages of these drugs. The one thing that's not discussed often, but which is really, really important, it's probably the reason why people die from taking these drugs, is hyperthermia. Temperature goes way up in individuals who take these drugs. And that usually happens between, after 4 hours, 4 to 16 hours, the temperature can go up. And depending on the dose, when people end up in emergency room, not only do they have hypertension and they might have had a stroke, but they really have very high temperature. And with high temperature, that's going to impact the function of not only your brain, but other organs, your kidney, and cause damage to your system.
All right. And then, so methamphetamine acts by increasing dopamine, norepinephrine in the synapse. And my understanding is that, a little basic synaptic biology here. When a neurotransmitter is released from an axon terminal of the synapse, it's important that, after it binds to the receptor on the postsynaptic cell, that it's rapidly removed.
Right.
And then that prepares the system to receive another impulse. So these drugs like methamphetamine, they inhibit the removal of the dopamine from the synapse. So there's more dopamine at the synapse for a longer time period, right?
Okay. So, but cocaine does that. So cocaine will block the uptake of dopamine or norepinephrine and serotonin too back into the terminal area, right? But methamphetamine and amphetamine they do something else in addition.
Okay.
They'll get into the terminals, they'll get into the vesicle that contain norepinephrine, dopamine, and they'll cause a release of dopamine into the terminals that get released into the synaptic area, right? Into the cleft. And in addition to causing the release, they'll also will block the reuptake of the monoamines, the catecholamines, back into the terminal to get metabolized. So when you use cocaine, let's say you were to use cocaine or methamphetamine equivalent dosages, you end up, and then we've published some papers on the release of dopamine in the past and showed that the methamphetamine will release a lot of dopamine and keep the level of dopamine in the synaptic cleft longer than cocaine. So it's really a double whammy, if I can use that term, release and blocking of the dopamine. So the dopamine doesn't get back in to get metabolized by those enzymes that will break it down. So it's really...
You've studied, one important area you've worked on is not only that methamphetamine, affects the function of the neural circuits by increasing dopamine at the synapse, but it also has adverse structural effects. There's actual damage to the nerve cells, right?
That is correct. So actually, when I first started to study methamphetamine, that was what I was the most interested in. Yeah. The reason why I was the most interested in this is because I came from the background of studying Parkinson's disease. Yeah. So Parkinson's disease, as a neurologist, involves the destruction of dopamine cells in the substantia nigra pars compacta, and that causes like a low level of dopamine, and then patients come in with tremor, they're slow, they cannot, they have problems walking, they might have some tremor. So because I was really interested in Parkinson's disease, and I started to be interested in what methamphetamine does to the brain, because there was some evidence that it was toxic. So I spent a lot of time trying to understand the mechanism of toxicity of methamphetamine. So when we give various dosages of methamphetamine, we documented that methamphetamine cause depletion of dopamine, cause loss of structural integrity of the dopamine terminals in the dorsal striatum, which is the area in Parkinson's disease where you have the low level of dopamine. So there's low level of dopamine in there. And the terminal degeneration occurred in terms of, you can look at markers of terminal integrity. So you have decrease in TH, okay? Decrease in dopamine transporter, which is the terminal area where the dopamine is taken back into the terminals. And then we have markers of the vesicles where dopamine gets sequestered before it gets released. And those markers are also decreased after methamphetamine. Now, our lab back in the 1990s showed, in addition to having degeneration of the dopamine terminal, you can have degeneration of serotonin terminals. And most importantly, we found evidence that dopamine can actually kill cells, non-dopaminergic cells, not only in the striatum, but in the frontal cortex and the hippocampus. So our lab was the first lab to show that. So before that, a lot of people say, well, there's no evidence that methamphetamine can kill cells, but we actually showed it that it killed neurons. But in addition to the neurons, there's evidence that methamphetamine can cause increases in the expression of glial cell. astrocytes, right? And which are also called astroglial cells, and microglial cells. All of these are increased in the brain of animals that get treated with methamphetamine. In 1998, we published a paper with a Japanese group showing that in the brain of methamphetamine users, there's also evidence of increased microgliosis in the brain of these individuals. So that's related to neuroinflammation, something that you're very interested in. And so if you have inflammation, that can lead to the release of oxidative compounds like oxygen-free radicals, hydroxyl radical, some nitric oxide. So we also have find evidence that this was the case with methamphetamine. And using an animal model, a transgenic animal model of that overexpress superoxide dismutase, which is an enzyme that breaks down superoxide radicals that are toxic, we show that those mice were protected from the toxic effects of methamphetamine. So we were the first one to show that. That paper has been cited many times. And other groups have been able to replicate the fact that methamphetamine does cause increased production of these free radicals in the brain.
And in patients with chronic use of methamphetamine, do they exhibit some clinical features that are similar to Parkinson's, or they certainly have problems with cognition, right? So I guess what I'm saying is, have you been able to relate the location and extent of the degeneration to actually the
clinical presentations? So there right now are 3 papers that I'm aware of. There might be others where people have actually documented that patients, individuals who have abused methamphetamine over a long period of time can present with Parkinsonism. It's not Parkinson's disease, but it's Parkinsonism. They have symptoms that are similar, not as bad, but similar to those that neurologists see in Parkinson's disease. Usually these people would come to neurological clinic complaining they might have symptoms, but it turns out they had a long history of using methamphetamine. Now, but those usually happen in people who are in their 50s. Now, the biggest problem from my perspective is the cognition, that because the cognitive abnormalities, deficits can happen in younger people, right? So that can impact their activities of daily living, whether they can keep jobs, whether they can follow instruction if they were to get involved in a treatment program. So the suggestions that I've made together with some other people is that when we're treating individuals with methamphetamine use disorder, we need to take into consideration the cognitive aspects of the disorder. So we need to come up with treatment that improve their cognition so that they can participate in other aspects of their treatment programs. So hopefully in the future, that's one of the things that we'll be doing. Treat the cognitive deficits and then involve people in programs that take care of some other issues related to the addiction per se.
So methamphetamine is clearly having adverse effects on neural circuits in multiple brain regions. You mentioned the hippocampus, which is like the brain region most people think about when they think about learning and memory and cognition. But also, I think, what about prefrontal cortex, which is involved in decision-making and kind of goal-directed behaviors?
Yes, the prefrontal cortex, as I mentioned a little bit earlier, when in the rats, when we look at toxicity, we were able to document toxicity at the level of the frontal cortex also. And then we can talk a little bit later about gene expression changes. But in papers, not from my group, from other groups, they've been able to document that not only do patients with methamphetamine users with methamphetamine use disorder have cognition, memory, and learning deficit, but they have difficulty with decision making using various tests. So they have difficulty with memory, this problem with decision making. So that's the reason why it's so important to address the cognitive aspects of addiction whenever you see a patient, not just drug-taking behavior. A lot of times we focus on the drug-taking behavior, not paying attention to the neurological aspect, right? Yeah. As a neurologist, I want more people to pay attention to the neurological aspects of addiction, because that's probably one of the reasons why people don't do well. So you can maybe reduce the drug-taking behavior, but how do people function? Because cognitively, if they're impaired, reducing their drug-taking behavior might not be enough.
This is a good point for you to tell our listeners about your interesting rat model, where you expose, say, a group of rats to methamphetamine, and then you're able, through some other manipulations, to identify some animals that are more prone to being addicted. Is that right? And others
yes,
not,
we do.
then...
Yes, that's a really good way of putting it. So what we've done, so earlier we talked about the fact that not everybody who abuse drugs meet criteria for substance use disorders. In the past, a lot of investigators would focus on all of the animals that self-administered drugs, right? As if all of the individual animals were the same. But so it turned out that in 2004, I have to admit, that was the first group that wrote a paper about cocaine, showing that you actually could use different manipulation to separate rats that were self-administering cocaine into animals that would continue to compulsively take the drug and other animals that would stop. So a few years ago, we wanted to know if this were the case for methamphetamine self-administering rats also. So we taught the animals how to give themselves methamphetamine. And by the way, rats really like to give themselves methamphetamine. So after they give themselves methamphetamine, and all of the rats will give themselves about the same amount of methamphetamine. And they start low, and then just like human, they progressively increase their intake of methamphetamine over time. So when they get to a certain level of stable level of drug self-administration, we say, okay, can we then separate the rats into animals that are addicted and animals that are not addicted, right? So the way we did that was using a foot shock, okay? And foot shock, so if 50% of the time when the animals press a lever in a cage to give themselves methamphetamine, they would get a foot shock. And what we did was we increased the foot shock over time to see if the animals would still go and press a lever, okay, to give themselves methamphetamine. So some rats will continue to take the methamphetamine despite the foot shock. So we call those adverse consequences. Now, that's one of the criteria in the diagnostic statistical manual, the psychiatric manual that diagnose methamphetamine use disorders. So we're able to create that criterion in rats. So in the rest of the rats, as you increase the food shock, they basically decrease and stop their intake of methamphetamine.
So this is like, so a person that, so this is kind of differentiating a person who they take methamphetamine and then they realize it's starting to have a negative effect on their job performance or their, interactions with their partner. It's having negative effects on their life, but they still keep taking the drug. So that's kind of similar to the rat, keep taking the drug even though it's getting a foot shot.
Yes, that's correct. I mean, the analogy I like to take is in terms of the food shock, when you get really high level, is somebody who's been arrested, he gets involved in the legal system, they put them they're in prison, they might be in prison, but they still want to have access to the drug, and they might continue to take the drug even in prison. And that happens, and it happens, everybody knows it happens. And the question is, how did the drugs get into the prison? So that's another story. But there are people, if they're using drugs, their parents find out they're using drugs and say, "Oh, we're going to cut you off." Or if they're in college, they're taking drugs and they know now they have to go have a job and be a productive, quote unquote, member of society, they stop drinking. They stop taking amphetamines, they stop snorting cocaine. So now they switch to, okay, I'm going to be a normal citizen, if you can use that term. But there are people, no matter what, they will continue to take drugs. Those are the ones who meet the criteria. Okay,
so now you have these rats in two different groups, and now you can look at their brains, right? So maybe this is a good point to talk about what's going on at the molecular level that might explain the differences between those two groups.
Yes. So what we did then, so we talked a little bit earlier about the different brain regions that are involved in addiction. So we have the nucleus accumbens, we have the dorsal striatum that represent habit formation, we have the prefrontal cortex that involved in decision making, and we have the hippocampus that's involved in learning and memory. So the first paper that we decided to do was to look at the nucleus accumbens, which is the rewarding, where the rewarding, quote unquote, aspect of the drug really seen, right? So we did a study where after we separated the animals, we dissected various brain regions, but we focused on the nucleus accumbens to look at epigenetic changes that might happen in the nucleus accumbens. Now, epigenetic is really very important for addiction because just gene changes might not be enough, because you need the way addiction happens, the way the behavior shows up, that even after a long period of abstinence, people might still crave the drug. So there must be some epigenetic reason that there might be some epigenetic changes that are persistent in the brain that impact why people want to take drugs.
And also Jean-Lud, are the rats you're using inbred? So essentially they have the same, there's no differences in their,
they're outbred, but
they are outbred.
Okay. They're outbred. So we wanted to replicate. Some people have said we should study some inbred rats, but I think from the perspective of humans are outbred, right? Yeah. So we wanted to study outbred. So it's like if it's happening in human, you cannot say, okay, all humans are monozygotic or something. They're very similar. So the inbred rats with specific non-changes would be interesting to do. But we thought doing ADBRED would be the way to start.
Okay, so I did have, when I talked to Eric Nestler, he talked a lot about epigenetics. He's another scientist doing a lot of work on addiction at the molecular level. But could you just kind of define what do you mean by epigenetic?
Okay, that's a good one. So epigenetic is defined as the interaction of genes and environment that bring individual phenotypes into being, right? So it's like you have heritable changes. A lot of time people talk mostly about genetics, but there can be heritable changes, right? So you can go from parents to children. So things that look like they're genetic, but they're not necessarily genetic. You can have heritable changes in gene expression that cause phenotypic changes, and there are no genetic changes in the DNA. So because the DNA can be modified without any really sequence changes, right?
Yeah.
So these changes are seen after histone modification. You can see changes in DNA modification. And there are a number of modifications that people have written about, and we've studied some of them. But I can list, for example, for the DNA modification, the DNA can get methylated. So you add a methyl group to the DNA, but the sequence of the DNA didn't change. It's just that on this one of the C-terminal, there's a methyl group attached to it. So there are no real genetic changes. And then you can have DNA hydroxymethylation. So now you have there's an hydroxyl group on the methyl. And then there are other modification that can happen on the outside of the bases. And then you can also have histone modification. The histones are protein, right? So you can acetylate the protein, you can methylate the protein, you can put a phosphorylation group on the protein, but there are no genetic, no strand differences between the animals that were treated before they were treated or after they were treated, because the methamphetamine is not causing genetic changes, but it's causing epigenetic changes. Yeah. So the interaction of the methamphetamine with the molecules can change some changes on the molecule, but no changes in the DNA sequence. So and that's epigenetic.
So these epigenetic changes, ultimately, they're going to lead to a change in the amount of messenger RNA encoded by the protein.
That is correct.
Sorry, encoded by the gene.
By the genes.
Amount of protein produced
right.
from that messenger.
So that can happen, right? So if the other thing about for example, if we did changes in the DNA methylation, you can cause either increases in the gene expression of the gene that code the protein. So if you have an increase in the expression of the gene, you might have more protein, or you can have a decrease in the expression of the gene that code a certain protein, so you have a decrease in that protein. So the other thing that can happen with the histone modification, the histone, when they get modified, they can bind to a normal DNA and cause changes in gene expression up and down, depending on what the histone, the modification of the histone. So I think in terms of cancer, neurological diseases, some psychiatric diseases, I think in the future, that's where a lot of the efforts need to go, because just looking at the gene sequences, the changes in gene sequences might not be enough to explain the interaction between the environment or an individual like you or me. So the consequences are going to be epigenetic, not necessarily genetics.
Okay, and then there are enzymes that either add or remove the modification of the DNA or the protein. So they either add or remove a methyl group, or they add or remove an acetyl group, right?
That is correct. So we have enzyme, for example, the one that modified the histone. They have histone acetyl transferases. Okay, so they'll put us acetyl group, or we have histone methyl transferases. So they are the methyl group. For DNA modification, we have DNA methyl transferases, or DNMTs. They add methyl group to the DNA, or we also have other enzyme called 10, 11. They take off methyl group, or they add hydroxyl group on the DNA. So there are lots of enzymes, and we've studied some of them trying to figure out, okay, what does methamphetamine do to this enzyme? What does methamphetamine do to DNA methylation or hydroxymethylation? What does methamphetamine do to histone modification? The reason why we're doing this is because it's, I think, in terms of just for the treatment, it's going to be very important to identify these epigenetic changes, because instead of waiting at the end, oh, there's this one gene that's affected, we're going to go after it. If we can document which histone modifications there are, or which DNA modifications there are, we can attack the changes at their source, right? So we can prevent, or we can attenuate, we can treat addiction where the molecular changes occur. So that's what I'm hoping, that we get to a point where we can actually do that with the, because it's really very difficult to treat addiction just focusing on one agent, a pharmacological agent that really impact one neurotransmitter system. So
having said that, Jean-Lud, in reading your work, one of the I guess classes of genes and coded proteins that seem to be different in terms of epigenetic modification of expression between the addiction prone and those not prone was potassium channels. Is that right?
Yes, that is. Yeah. Potassium channels are very, very important. And because you've been doing the neuroscience work for such a long time, potassium channel, we're very excited about potassium channels, even though we're saying the epigenetic story, but the potassium channels are very interesting because we identified the potassium channel via a epigenetic study, right? So what we did was we take the nucleus accumbens, we process the tissue, and we look at DNA hydroxymethylation. The reason why we pick DNA hydroxymethylation is because there are a lot of papers, including papers from Hopkins, showing that hydroxymethylation plays really very important roles in the brain. So we say, maybe that's true for methamphetamine also. So we identify a number of genes that are impacted in terms of increases in DNA hydroxymethylated peaks or decrease in DNA hydroxymethylated peaks. And a class of gene, it wasn't just one of them, it's like multiple potassium channels were showing increase in DNA hydroxymethylated peaks. So you say, wow, this is really interesting that there is a whole class of genes that are impacted. So we then look at the gene expression for the potassium channel, and we saw that there were increases in the expression of the potassium channels in the animals that suppress their intake of methamphetamine. So that actually made sense, because if you overexpress potassium channel, then you're going to get hypopolarization, decreased excitation, and decreased release of neurotransmitters. So say, wow, this is really perfect.
Yeah,
So
their neurons are able to kind of quiet down after being exposed to the methamphetamine and maybe quiet down and then, you know, if this is occurring all over, say, for example, prefrontal cortex,
yep,
then they'd have better control over their behavior.
that's exactly what we're thinking.
And these are
And
druggable. I mean, there are, let me tell you a story, Jean-Luc. When I
okay.
was at University of Kentucky, so this is in the 90s. So there's a drug called diazoxide
Mm-hmm.
that opens potassium channels. It actually used to be used clinically to treat acute hypertension
Mm-hmm.
because it's very, at higher doses, it's very powerful.
Yeah, it's very potent, yes.
But at lower doses, we did these studies and actually continued at NIA where we found in Alzheimer's models where we think there's a cytotoxic process going on
Mm-hmm.
where the neural networks, we've found it was beneficial. But at Kentucky, there was a clinician there who worked with Alzheimer's patients. And, you know, we tried to get a company to support a clinical trial, actually, but it was off patent and they weren't interested in it because they're not going to make much money from it, even if it works. But he just, this is something he could prescribe to patients, he gave Alzheimer's patients low dose, these early Alzheimer's, low dose diazoxide and found some beneficial effects. So I think that a low dose, something like that could actually be tried with methamphetamine.
So what we're actually doing right now actually is writing a protocol for rats first. So we're going to try diazoxide. There's some other potassium agonist that we're going to try in the rat model and see what happens. If those work, then we're probably going to go to human, try to convince. We don't have enough meth users in Baltimore. So maybe we can convince some physicians who are seeing people in other places to.
That's interesting. What,
yeah.
you know, Baltimore, what's the explanation for that?
I don't know. I mean, when I was doing a lot of clinical research, I tried to recruit meth users from this area, but it wasn't really easy to do. So I think heroin, if you want heroin users in Baltimore to study, you can find. If you want to use cocaine users, marijuana users, are we able to recruit people from those patient populations? But meth users, it was impossible to get. You can get MDMA, even MDMA.
But this potassium channel approach could also work with cocaine, right?
Yes, we're hoping that's the case because the same process should be. We actually wrote a review of the potential use of potassium channel in substance use disorders that was published, I guess, last year, where we look at a lot of the different alcohol, methamphetamine, cocaine, and to see, and then hopefully people would have read this and then try it in their cocaine models or even in human, because these things are available clinically. So
now, one thing I saw that was pretty intriguing was this that you did was this methamphetamine preconditioning, where so the idea here it's something we call hormesis. Actually, it's cells, including nerve cells, respond adaptively to a mild stress so that they become more resistant to a severe stress. And this kind of is a very important concept in the standpoint of neuroprotection. You know, where you have a condition where, as you've shown with methamphetamine, where neurons are degenerating. And so I was wondering, what about preconditioning? Like, you know this, we've done all this work with intermittent fasting or exercise. does exercise have any impact on drug addicts if you can get them into an exercise program?
Actually, there are treatment programs that have incorporated. Haven't done that, but there's treatment programs that have incorporated exercise in their treatment programs.
Yeah.
Then the evidence we actually co-authored a paper with a group from France where they have rats exercising and other rats not exercising, and show that the animals that exercise are less likely to take self-administered drugs. So there's evidence that exercise does work. So what we actually want to do in the future is actually work together with them, the French group, to look at the epigenetics and the potential, like going forward. But we've actually tried the hormesis, the preconditioning in the METH toxicity model, which is different from the model that we're using now. So if you give low dosages of methamphetamine over time, not toxic dosages, but low dosages that don't cause major damage to the brain of the animal, and then you expose them to a larger dose of methamphetamine, you can protect them from the large toxic dose. I think one of the things that we thought at the time was that that's why more people who take methamphetamine don't present with large scale Parkinson's disease, because they started out with low dosages and then they progressively, some of them, people who are susceptible, maybe they have low enzyme, superoxide dismutase, low GSH, glutathione, low other antioxidant, they're not taking enough vitamins, they might be more prone to develop toxicity and have Parkinsonism.
And you found with that methamphetamine preconditioning that you saw increase in levels of brain-derived neurotrophic factor.
That's correct. BDNF was increased.
And that's seen with exercise for sure. Exercise will cause a big boost to BDNF in the brain. Right. We found fasting can have not as much as exercise, but it can increase. So maybe that's a kind of a common mechanism in neuroprotection.
Well, I think BDNF is playing a big role in the neuroprotection. I mean, there's like, it would be tough to do that kind of study in humans. Okay, but but one might want to look at the periphery, the blood periphery, right? In blood, and see, okay, do they have increased level of BDNF or something?
Cerebral spinal fluid, if you can.
CSF, you could do that. But a lot of people don't want you to do spinal taps on them. So that's a tough
No,
one.
I know. Yeah. Okay, to finish up here, so this morning I went, so there's emerging evidence that psychedelics can benefit people with certain addictions. I think there's been, even going way back to the 50s or 60s, some work with alcohol addiction. Now, Johns Hopkins on the Bayview campus where you are, and actually just a stone's throw from where you are, they started a Center for Psychedelics and Consciousness Research.
Right.
Now, I don't like the last part of that name, the consciousness research. Isn't any research being done? Hopefully it's on conscious people.
Yes, I guess that I think the reason why they have consciousness there, I'm guessing, I don't know, it's because, like, back in the days, the people were using these drugs, were using it to expand their consciousness. Then, they're using psychotherapy and all that other stuff. But this is not something that I've studied. I actually have no interest in really studying psychedelics as treatment for methamphetamine. My hands are right now totally full, and I will leave it to the Hopkins people they seem to be doing a really great job of it. I think there was another group, I cannot remember exactly, that also getting into the doing those studies.
I think maybe up in New York or something.
Right. So I think it looks promising for them to do. But I mean, people are using ecstasy, for example, MDMA for PTSD, and then post-traumatic stress syndrome. So all of that's going on, and then then what would be interesting if it really pans out in humans is try to figure out the molecular mechanism for this. But I don't really have time for doing that. Methamphetamine is really very interesting. There are a lot of stuff we're finding out with methamphetamine. There are a lot of genetic manipulation we hope to be able to do in the future to try to understand this, because there are a lot of people who suffer from methamphetamine use disorders, and we need to come up with a better way of taking care of our patients, of our American people. We need to do that.
I want to mention one method. I think you've probably heard about it. Dimitrios Kapagiannis, neurologist.
Yes.
Who was in my lab when I was lab chief. So he developed this method back up a little bit. So all cells release these very small membranous vesicles called exosomes, or extracellular vesicles. And so if you take blood from a patient, or take blood from anybody, there's a method to remove these small vesicles from the blood. Some of them are coming maybe from the vascular endothelial cells, some may be coming from the liver, and some seem to be coming from the brain. And Dimitrios developed a method to take the total pool of these vesicles in blood, and using an antibody against a neuron-specific protein, pull out the vesicles that are coming from neurons. Now, these vesicles have a lot of proteins, they have messenger RNA, and he's kind of uses this as develop biomarkers for mild cognitive impairment and Alzheimer's. But this can also be used in basic science studies, like even in humans. So down the road, somebody may take someone in human patients with addiction, or maybe some ones who are resistant to addiction, and then isolate vesicles coming from neurons. And then you could even look at, maybe epigenetic changes even.
No, we've actually considered it. And then because, I know him, and, I've interacted with him, and I've listened to some of his talk, and I've read his papers, and it's really actually a good way. We talk just a little bit about measuring BDNF and stuff like that in the brain of people. One of the reasons why I considered this was, wow, since we cannot get biopsies of the brain, you might be able to just isolate neuron-specific vesicles and see what happens in there. You can do proteomics, right? And then we look at a lot of different proteins.
This is being used to diagnose cancers now. And in fact, the cancer people were the first ones that studied these vesicles, because it turns out that there's molecular fingerprints of vesicles coming from cancer cells that distinguish them from vesicles coming from normal cells. And so they actually coined the term liquid biopsy.
Yeah, but that's perfect, actually.
Yeah. Okay, well, I think you've covered a lot of ground. We've been talking for an hour and 40 minutes. And I'm going to put in the description section of the videos on YouTube links to some of your review articles, relevant to methamphetamine, and maybe a link to your lab page or something.
Yeah, that would be great. And then also a link to the potassium channel article, because I think that's a really good way of I think that potassium channel story is really interesting.
Yeah, I think it's very exciting. There's a lot of potential there. There's, as you mentioned at the outset, there's no treatments right now that will get people off. It's in opioid addiction, there's at least one treatment that seems
Right,
to help a lot,
exactly.
buprenorphine. And although apparently I read somewhere that it's being, at least in some places, being underutilized.
Oh, it's actually being utilized quite a bit, and there's quite a few controversial statement that's been made about that one, because especially with regards to African American patients, because they don't have access, they haven't been able to, and then it's difficult to get doctors who are prescribing. So there are a lot of issues related to that at some point or another, somebody will have to deal with. Yeah. Because, if heroin users who are African-American don't have access to treatment, that's terrible. Yeah. It's a better drug than methadone by all accounts. So it is. It can be an issue.
And it's not an expensive drug. So it's just a systemic problem.
It is. I mean, so there's the papers saying that pregnant women who are heroin users, opiate users, don't have access. I mean, there's a whole literature.
And then, and that, it's adverse effects on the brains of their developing baby if they're taking these drugs when they're pregnant, right?
Yep.
Huh. Wow. What about, is that true with methamphetamine and cocaine too? If a woman's pregnant taking those, do those have adverse effects on the?
So with cocaine, I don't know if you remember this. There was that whole brouhaha back, I guess, in the 80s and early 90s about cocaine babies. Okay. And how developmentally they were going to be totally, it's a lost generation. It didn't turn out to be that way. So I think the jury is still out in terms of what really happens as these, I guess most of these people are like in early 20s now, but the clinical manifestation that people were seeing and making these predictions based on those didn't really pan out.
That's good.
Yeah, that's very good.
Yeah. Okay, Jean-Lud, it's great to see you again, and I'll try to keep in touch now and then.
Yes, it's great. Keep me posted. Let me know if you need anything else, and then good luck with your nice retirement. You're looking really, really, really good for a retired professor.
You're looking good too.
Thank you.
Okay, Jean-Lud. Bye.
See you later.